Hello, am I audible? And can you see my video? Okay, thank you. Please let me know if the presentation is visible to you. Okay, thank you. So, very good evening. We will join. We will start our class soon once more students will join. Till then, please wait. Okay, and let me know if there is any problem with the voice and also with the PPT and my video. Okay, so it's 6 p.m., but I will wait for 1 or 2 more minutes till other students join. Then I will start with the introductions. Let's wait for 2 more minutes. Okay, let me start with the introduction at least. So, very good evening to the students who have joined. My name is Mousumi Deb, and I am the PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am the TA for your NPTEL course of the Medicinal Chemistry, and today it is our week 8 live session. So, in every week, we are having this kind of live sessions on Tuesdays from 6 p.m. to 8 p.m., where I discuss various topics and various questions related to that week's content. So, since we are in the very first lecture, we had completed week 1 and week 2 all together. So right now we are going ahead with every week in every each class. So like today it is the week 8 live session, but the topics we will discuss it from the week 9 course content. Right now the week 9 of the course medicinal chemistry is live, I believe, and we will today discuss around 20 questions related to that week 9 course content. All the questions will be MCQs, and this kind of question will help you to answer your weekly assignments, and also when I am discussing the questions answers, it will help you to understand the course contents better. Okay, so meanwhile when other students join, I will clarify few points which I repeat every before every class. So I hope my voice is audible and the video and the PPT is visible to you. If not, you can kindly let me know in the chat box. Okay. So yeah, so there will be this kind of problem solving sessions every week. Okay, and the sessions will be live on Google Meet platform. The joining code is same for every week, and we will have this similar the same joining code for the Google Meet link for the rest of our course. Okay, and these sessions will be on every Tuesday from 6 p.m. to 8 p.m. So the important point is, if any kind of network disruption happens from my side, it can happen from my side and from your side also. If it happened from my side, so please do not leave the session. Okay, I will try to join as soon as possible, but all the students are requested to not leave the class, because I will try to join as soon as possible. And during the session, if you have any kind of your doubts and query and questions, you can post it via the chat box. Okay, because of the for the smooth streaming of the class, I have just muted you all and kept the videos off, so that I can record the session very easily. Since I am recording the session, the sessions will be this recorded video, and also the PPT will be shared to you soon. You can find it on the swam portal. If you go on the course content and assignment page, in the below you will find a Google sheet containing the problem solving sessions, where the video link and also the lecture, the PPT link is provided to you. So you can just check it from there. If you miss anything during the session because of the internet connection, so you do not have to worry. If there is any network disruption from your side, you can rejoin at the class at any time, because I have kept it open. So you can just join at any time, there is no problem. And during the session, since I have muted you all and kept your cameras off, but the chat box is open. Okay, I'm checking the chat box now and then every time. So if you have any doubt, any query, you can just post it on the chat box. I will check it later, and then I will just discuss it with you soon. And if there is any doubt to persist after the class, you can just drop an email to me. I will surely answer your doubts, your query after class also, because at the end, always I share my email ID. So if you have any problem, any doubt, you can just mail me about your problem and doubts. And as I said, this presentation and class recording will be shared to you soon. Okay. So today, as I have said earlier, today it is our week 8 live session, but the content which we will discuss today will be from the week 9 course content. Okay, so I think I don't know if other students have joined or not. Okay, but I will start my session because I have only 2 hours to complete the contents which I have prepared. So let's just start the session. So before starting, let's see what is in your week 9 course content of the course of medicinal chemistry. So the course contents are as like this: the optimizing access to the target, prodrugs and drug alliances, endogenous compounds, peptidomimetics, oligonucleotides as drugs, and there will be a tutorial of optimizing access, which is the prodrug. Okay, so let us see what is the first question of today's session. So yeah, the first question of today's session is, which of the following can act as isostar of hydrogen? Your options are number A, nitrogen, sodium, number B, oxygen, number C, fluorine, and number D, nitrogen. So which of the following can act as an isostar of hydrogen? In the previous lectures, I have discussed about what are the isostars and also what are the bioisostars. I have discussed it in the previous class. So today I will also give a description about this bioisostar and isostar topic, because it is a very important part of the prodrug design. Okay, let me check the chat box. Okay, so I have one answer which says it is A. Okay, now let me show what is the correct answer and let us discuss about it. So the correct answer will be, the following which can act as an isostar of hydrogen is number C, fluorine. So now bioisostars. So the development and application of bioisostars have been adopted as fundamental tactical approach useful to address a number of aspects associated with the design and development of the drug candidates. The utility of bioisostars extending to improving the potency, enhancing the selectivity, alternating physical properties, reducing or redirecting metabolism, eliminating or modifying toxicophores, and acquiring novel intellectual property. These are the utilities of the bioisostars. As well, in the last day, I also have discussed about few about the bioisostars. That it is a fundamental tactic which can be used to improve a drug design and development. In many times, there are in a particular lead compound, you will see there is a groups, some groups will be important for the biological activity, but the similar groups will be responsible for the metabolic instability of that compound. So in that cases, that particular group should be replaced by a bioisostar of the similar group to retain the similar biological activity, but to improve the potency and to improve the selectivity or the metabolism of that particular drug. So like fluorine as an isostar of hydrogen, the unique properties of fluorine have led to its widespread application in drug design as an isostar of hydrogen. Since incorporation of fluorine can productively modulate a range of properties of interest to medicinal chemists. Like the fluorine, as you can see the hydrogen, the van der Waals radius is 1.20 angstrom. But in case of fluorine, that van der Waals radius is 1.35 angstrom, which is very similar to that of the hydrogen. So the hydrogen and fluorine, there won't be a very high structural difference if we replace the particular hydrogen with a fluorine, right? So that's, for example, there is said that 20% of all drugs are fluorinated compound. Yes, because the fluorines are very good for the biological activity of the drugs. There is a unique property only, actually from the halogens, there are only fluorine is such a halogen which is a very good biological activity rather than chlorine and bromine and other heavier halogen atoms. But the fluorine has some unique biological property and it is very similar to that of the hydrogen's structure. So we can replace any hydrogen with a fluorine to improve the potency, to check if if it helps the particular activity. Okay, so then uh, now, replacing a group in the lead compound with an isostar makes it easier to determine whether a particular property such as hydrogen bonding is important. We'll look at several examples. One way to do is to replace with a methyl group because it has the same valency, and fluorine, which we discussed it often as an isostar of hydrogen because because it has a similar size and so it is more electronegative. It can be used to vary the electronic properties of the drug without having major steric effect. So fluorine is very commonly used as a star in this concept. Okay, so the correct answer was fluorine for the question number 1. So now I will move forward for the question number 2. So problem number 2 says D1927 contains a branched alkyl group shown in the blue, which protects the terminal peptide bond from hydrolysis. You can see the structure from here. This is the D1927, which is a branched alkyl group which is shown in blue, this one, and it protects the terminal peptide bond from hydrolysis. This is a terminal peptide bond, okay, for this drug. What is a general term given for such kind of group? Your options are number A, stereoelectronic shield, number B, electronic shield, number C, steric shield, and number D, alkyl shield. Okay. So what is the particular term given for this kind of group which protects the terminal peptide bond from hydrolysis? Stereoelectronic shield, electronic shield, steric shield, or alkyl shield? I will give you some time. Think, look at the structure very carefully and then answer this question. D1927 contains a branched alkyl group shown in the blue which protects the terminal peptide bond from hydrolysis. What is the general term given for such kind of group? Stereoelectronic, electronic, steric and alkyl shield. Let me check the chat box. Okay, so I have different answers. Somebody says the number B and then number C. Okay, let me check which of the following, number B or number C, is the correct one. So the correct answer will be number C, steric shield. Now let us look at what is a steric shield is. So basically, steric shields are some functional groups. It is used in case of some functional groups that are more susceptible to chemical and enzymatic degradation than others. For example, esters and amide are prone to hydrolysis. A common strategy is to protect these groups to add a steric shield designed to hinder the approach of a nucleophile or an enzyme to the susceptible group. These usually involve the addition of a bulky alkyl group close to the functional group. For example, tertiary butyl group in the anti-rheumatic agent D1927. It serves as a steric shield and blocks the hydrolysis of the terminal peptide bond. Okay, so the steric shields, are the rationale of using the steric shields are, these are used to increase the chemical and metabolic stability. Okay, then introduce bulky group as a shield, protects the susceptible bonds such as ester, amides for hydrolysis. It hinders the attack by nucleophiles or enzymes. So as we all know, this is the terminal amide bond and this is a peptide bond and it is the anti-rheumatic agent D1927. So this kind of terminal peptide bonds or amide bond, these are prone to hydrolysis by enzymes or various kind of reagents like nucleophiles. But if you use this kind of tertiary butyl, which is a very bulky group here at the terminal end, it will block that particular hydrolysis of the terminal amide, and this is known as the steric shield. Okay, so this the correct answer for this question was the number C, which is the steric shield. So now I will go to the next problem. Anyways, if you have any doubt, any query during the class, as I have mentioned always, you can just let me know in the chat box. Okay, for the smooth recording of the class, I have muted you all and kept the cameras off. But if you have any problem, any doubt, you can just let me know anytime in the chat box. Okay, I will repeat that particular question or repeat that problems which you have which you mentioned in the chat box. Okay, at the end of the session. So please feel free to let me know what is your problem and if you do not understand the point. Okay, so this was the problem number 2. So now I will move toward the next problem. So problem number 3, it says that why should the introduction of an ester group into the main skeleton of a drug decrease the drug's duration of action? Your options are: number A, it makes the drug more polar and increases the rate of excretion. Number B, it increases the molecular weight of the drug and reduces the amount of drug absorbed. Number C, it reacts with proteins in the body such that the drug is irreversibly linked to the proteins by a covalent bond. And number D, it is hydrolyzed by the esterases to an alcohol and carboxylic acid, which are quickly excreted due to the increased polarity. Why should the introduction of an ester group into the main skeleton of the drug decrease the drug's duration of action? Again, I will repeat the options. Number A, it makes the drug more polar and increases the rate of excretion. Number B, it increases the molecular weight of the drug and reduces the amount of drug absorbed. Number C, it reacts with the proteins in the body such that the drug is irreversibly linked to the proteins by a covalent bond. And number D, it is hydrolyzed by the esterases to an alcohol and carboxylic acid, which are quickly excreted due to the increased polarity. I will give you some time. Read the question, read the options very carefully, and let me know in the chat box which of the following you think is the correct option for the problem number 3. Since there is no marks given for answering into the class, but if you answer, I will understand if you are listening to me properly, or if you have understand that point or not. Okay, even if your answer is wrong, please try to answer. Otherwise, we won't have the fruitful conversation in the class. Okay, so please let me know which of the following you think is the correct one. And if you have any doubt, at any point of the class, please do let me know in the chat box. Let me check the chat box now. Okay, so I think I have few answers. Number B, number A, number D. Okay, so again I have different answers. I have three different answers for the problem number 3. Okay, so let me check which of the following is the correct one, and let us discuss about it. So the correct answer will be number D. It is hydrolyzed by esterases to an alcohol and carboxylic acid, which are quickly excreted due to the increased polarity. Okay. So see, esterases are such a group of enzymes which hydrolyzes the ester group. So we all know if the ester is hydrolyzed, what it will form? It will form acid and an alcohol, right? Because acid and alcohol, they together forms an ester, and esterases are such the group of enzymes which degrades this ester bond, and it forms the carboxylic acid and this particular alcohols. So is the same is for the polyurethane. If there is a polyurethane, they will be also hydrolyzed by this particular esterase to the alcohol and this kind of acids. Okay, and because these groups are because these carboxylic acid and alcohols, they are very polar, they will be quickly excreted. Okay, if you remember, I don't know if you remember or not, in last class I discussed about the your bioisostars of carboxylic acids, right? So in, if you just go and find that particular PPT which I have shared for the last class, which is week 7 live sessions PPT, if you check there, I have mentioned that carboxylic acids are not very good functional groups for a particular drug, okay? Because if the molecule contains a free carboxylic acid group, these they don't show very good pharmacokinetic profile because of this free carboxylic acid group. They are this makes the compound very polar, and they are excreted very soon. Okay, so that is why this particular carboxylic acid groups say we cannot just keep them free, okay, in the particular drug. We cannot keep them free, and that's why we have to use several bioisostars of carboxylic acid, like tetrazole. Tetrazole is a very important and a very popular bioisostar of carboxylic acid group, and that is the requirement of because of this kind of requirement, there is a lots of number of carboxylic acids bioisostars, okay? And if you go back and check that PPT, you will see that I have mentioned there are several groups which acts as a bioisostar of carboxylic acid, because it is a very common to find out a very important bioisoster of carboxylic acid, so that the biological activity is retained of the compound, but not but it should be metabolically stable, okay? So answer number D was the correct, that the introduction of ester group main skeleton of drug decreases the drug duration of action, because it is hydrolyzed by esterases to an alcohol and carboxylic acid, which are quickly excreted due to the increased polarity, okay? So now I will move toward the next problem. So problem number 4 says megestrol, which is acetate, has a short duration of activity since it is metabolized at the position number 6. This is the megestrol acetate, and this is the position number 6, which is mentioned in the blue. Placing a methyl group at the 6th position increases the duration of action. What role does the methyl group play? Okay, if we place a methyl group at the position number 6 of this megestrol acetate, it will improve the duration of the action of this particular drug. So what is the role of this methyl group, which if we add will play? Number A, it is susceptible to metabolic group. Number B, it is a metabolic blocker. Number C, it is a conformational blocker. Number D, it is an electron donating group. So which will be correct for the role of the methyl group? Number A, it will be a susceptible metabolic group. Number B, it is a metabolic blocker. Number C, it is a conformational blocker. And number D, it is an electron donating group. Magistrol acetate has a short duration of activity since it is metabolized at position number 6. Placing a methyl group at 6th position increases the duration of action. What role does the methyl group play? Number A, it is a susceptible metabolic group. Number B, it is a metabolic blocker. Number C, it is a conformational blocker. Number D, it is an electron donating group. Let me check the chat box now. Okay, so I have few answers. Number B, number B, number D. So I have two number B and one number D. Okay, let me check which of the following is the correct one. So the correct answer will be number B. It is a metabolic blocker. So metabolic blockers. Some drugs are metabolized by introducing a polar functional group at the particular position of the skeleton. For example, the megestrol acetate, which is an oral contraceptive. Steroid is oxidized at the position number 6 to give a OH of this position, but introducing a methyl group at the position number 6, the metabolism is blocked and the activity of the drug is prolonged. That's why this methyl group plays the role of a metabolic blocker, because it is blocking a particular metabolism process. Okay, and this increases the duration of the drug. Okay, so the methyl group at the number 6 position, it will act as a metabolic blocker. Okay, so now next question. Problem number 5 says salbutamol is an anti-asthmatic agent which shows an improved acidity over previous catechol-like compounds. What strategy was used in order to attain the improved duration of the action? Your options are number A, substituent variation, number B, group shift, number C, chain contraction, and number D, simplification. Salbutamol is an anti-asthmatic agent which shows an improved acidity over the previous catechol-like compounds. What strategy was used to attain the improved duration of action? Your options are number A, substituent variation, number B, group shift, number C, chain contraction, and number D, simplification. As you can see, this is the structure of the salbutamol. We have a secondary alcohol, primary alcohol, and phenolic OH, and then we also have a tertiary butyl group here. Let me know in the chat box A, B, C or D which of the following options will be correct for the problem number 5. What strategy was used in order to attain the improved duration of action for the salbutamol, which shows improved acidity over the previously catechol-like compounds? Okay, let me check the chat box now. Okay, I have only one answer for number C for this particular question. Okay, I will give you 2 more minutes. Others, please also respond. Okay, please let me know which of the following you think is the correct answer. Even if your answer is wrong, it doesn't matter. So, but please answer. Okay, I will give you 2 more minutes. Others also try to answer this question. Which strategy was used in order to attain the improved duration of action for salbutamol? The structure is given here. The options are given here. Just look at the structure carefully, read the options carefully, and then let me know which of the following you think is the correct one. Now let me check again if I have few more answers. Okay. Okay, let me show you what is the answer of this question. So, the correct answer will be number B, group shift. So, group shift is the strategy which was used in order to attain the improved duration of action for salbutamol. So, what is a group shift? Basically, removing or replacing a metabolically vulnerable group is feasible if the group concerned is not involved in important binding interaction of the binding site. If the group is important, then we have to use a different strategy such as either mask the vulnerable group by using prodrugs or shifting the vulnerable group within the molecular skeleton. Okay, but by this tactic, salbutamol was developed in 1969 from its analog neurotransmitter noradrenaline, which is have a catechol structure. But noradrenaline is metabolized by methylation of one of the phenolic group of the with catechol O methyl transferase. The other phenolic group is important for the receptor binding interaction. So, for example, here it is given like salbutamol, which is derived from the noradrenaline, where in a noradrenaline both phenolic groups are involved in hydrogen bonding receptor. Thus, metabolic methylation of one of the groups make the compound inactive, and it has a short duration. A solution is to move one phenolic group out from the ring by one carbon unit if more activity is lost due to the improper binding. So, if you can see here, so in case of particular noradrenaline, what it has a catechol-like structure. Okay, so what is the structure for the catechol? I will just draw it here. So, in case of noradrenaline, we have this catechol-like structure where two OH groups are joined with the group, like two phenolic OH side by side at 1 and 2 position. It is known as catechol, we all know. So, the noradrenaline has a catechol-like structure. You can see it from here. Okay, here it is mentioned. The noradrenaline has a catechol-like structure. Here, both of these OH groups are involved in the binding with the receptor. Okay, so but the one OH group, it is susceptible for the methylation by the enzyme catechol O methyl transferase. And if the phenolic group is methylated, so what will happen? The activity will be less. But in case of salbutamol, what happened? This phenolic OH group is shifted with one carbon. Okay, here the phenolic OH group is moved from the directly joined OH group from the phenyl ring. It is moved by one carbon. Okay, here and this will intact keep the binding of the with the receptor intact, but it will not get metabolized or methylated by the catechol O methyl transferase enzyme. So now what will happen? The activity of the compound will be increased. Okay, so that is a reason for the improved activity of the particular salbutamol from their precursors. Okay, so remember the correct answer will be group shift. Okay, So I hope you have understood. Anytime, any problem, if you have understanding, let me know in the chat box. Okay, I will repeat that question or that particular problem. I will just try to solve it. So now next question. So problem number 6 says, what sort of therapy involves administration of an antibody linked to an enzyme such that the enzyme activates the prodrug? Your options will be number A, ADAPT, number B, GDAPT, number C, ADEPT, and number D, GDEPT. What sort of therapy involves administration of an antibody linked to an enzyme such that the enzyme activates a prodrug? What sort of therapy involves administration of an antibody linked to an enzyme such that the enzyme activates the prodrug? Let me check the chat box now. Okay, I have few answers, number B, number C. Okay, let me check which of the following is the correct one. Okay. So the correct answer will be number C, ADEPT. It is the therapy which involves the administration of an antibody linked to an enzyme. So in antibody-directed enzyme product therapy, which is the full form of ADEPT, antibody-directed enzyme prodrug therapy, is an antibody-bound enzyme is targeted to tumor cells. This allows for selective activation of a non-toxic prodrug to a cytotoxic agent at the tumor site for the cancer therapy. Site-selective prodrug activation results in the reduced side effects in the remote tissue. So this, what is the difference between ADEPT and GDEPT? Okay, the in case of ADEPT there is a prodrug and which is of the drug which is cytotoxic. So now the cytotoxic drug which is given to the particular glioma cells, which are the cancer cells. So here is the systematic administration of antibody enzyme conjugation. So we have an enzyme, then we have an antibody which is the specific for that particular your tumor cell marker. Okay, so when there is an enzyme which is linked to antibody specific for tumor cell marker, it binds to an antigen. Okay, and the drug, it will this in the ADPT theory, the drug will be guided by this kind of antigen antibody interaction through the glioma cells. Okay, and in case of GDPT, what will happen? This direct injection of vector transgene into the brain tumor. Brain glioma cell is the brain tumor cell. Okay, so in case this is the target tumor, so what happens in GDPT? Target tumor specific transcription happens and gene encoding the prodrug here. Then we will have the transduction, and by the transduction, tumor specific foreign gene expression will activate the prodrug, and this will make which will give to the drug to the glioma cell. So all this prodrug to drug activation will be happen inside the your tumor cells. Okay, but in case of ADPT, it will be outside, and the drug will be directly go to the particular cell directed by the antibody enzyme antibody interactions. So this is the abbreviated form, remember, and antibody directed enzyme product therapy, it is the full form of ADPT. Okay, and for the GDPT, it is a gene directed enzyme product therapy. Okay. So now next problem, problem number 7 says which of the following will not be a limitation for a prodrug design. Number A, formation of the toxic product. Number B, an inert carrier can be cleaved off forming toxic product. Number C, product cleaving off before reaching the target site. And number D, the cleaving of the carrier also site specific. Which of the following will not be a limitation of prodrug design? Number A, formation of toxic product. Number B, an inert carrier can be cleft off forming a toxic product. Number C, product cleaving off before reaching to the target site. And number D, the cleaving of a carrier is also site specific. Which of the following will not be a limitation of prodrug design? Formation of toxic product. An inert carrier can be cleft off forming a toxic product. Product product cleaving off before reaching to the target site, or cleaving of the carrier is also site specific. Let me check the chat box now. Okay, I have few answers. Number B, number D. Okay, I have two answers, number B and number D. Let me check which of the following will be the correct one. So number B and number D is the given answers. So the correct answer will be Number D, cleaving of the carrier is also site specific. It will not be a limitation for the prodrug design. So what is a, let us first understand what is a prodrug and prodrug design. So how the product design can be happen. So prodrug is a pharmacological substance administered in an inactive form. Once administered, the prodrug is metabolized in vivo into the active drug within the body through the metabolic process such as the hydrolysis of an ester form of the drug. Sometimes drugs are designed to make use of metabolic process in order to generate their active form. This is done in order to improve some selected property of the molecule such as water solubility or the ability to cross the membrane temporarily. So I will make it a little bigger so you can just look at it carefully. So I think it will be now more easily, you can just look at it. So yeah, I will repeat. Prodrug is a pharmacological substance administered in an active form. Once administered, the prodrug is metabolized in vivo into an active drug within the body through the metabolic process, such as hydrolysis of an ester to form a drug. Sometimes drugs are designed to make the use of metabolic processes in order to generate their active form. This is done in order to improve some selected properties of molecules, such as water solubility, ability to cross a membrane temporarily. But what are the limitations of the prodrug design? The limitations of formation of unexpected metabolic from the total prodrug that may be toxic. The inert carrier generated following cleaving of the prodrug may also transform into toxic metabolite. During the activation stage, the prodrug might consume a vital cell constituent leading to its depletion. The limitation of product designing is the formation of unexpected metabolite, as I have mentioned here. The inert carrier can be cleaved off, forming toxic molecule, and product might consume a vital nutrient. If the carrier cleavage is site specific, then it is an advantage. Okay, so I will just summarize what is a product design. So sometimes it happens that we have to give a drug. Okay, let's say we have to give a drug orally. Okay, but sometimes the functional group which are in the drug are not suitable for the oral administration. Okay, or let's say we have given the drug via any other method of administration, but it will be not water soluble, which is required for our mechanism of action. So now what we will do is we will design a prodrug which will form the active form of drug where when it is into the system, the systematic circulation, or into the particular water medium or acidic medium of the our stomach or any kind of your liver or any kind of other organ. So then the product design, this it can be done like now, like this. Okay, but sometimes it can happen. What are the limitations of the product design? That whenever there is a this product formation, let's say for example we have the active form has the acidic side and we have made a ester to administer it. So now into the when it is given to the body, the esterases will cleave the ester to the particular acid and alcohol form. Now let's say that particular alcohol for the acidic form is important for our body, that is the drug, but the alcohol form which is formed from that particular acid, it can be toxic. Okay, so it is given here that formation of the unexpected metabolite which can be toxic. The inert carrier can be clipped off for forming a toxic molecule, and also it can consume a vital nutrient of our body during the metabolism. So that are the limitations of the product design. But if the cleaving of the carrier is site specific, then it is the not it is the not limitation. It is can be treated as an advantage of the product design. Okay. So the option number D was the correct for the problem number 7. So now I will move forward. So problem number 8 says L 791456 is an anti-arthritic drug. This is the anti-arthritic drug L 791456. Which of the following structures is not likely a metabolite? Your options are number A, this, number B, number C, and number D. So four structures are given. Okay, so which of the following structure is not likely to a metabolite of the L791456 drug? Structure A, B, C, or D? Look at the drug structure carefully, look at the options carefully, and then let me know which of the following structures is not a metabolite for L791456. L791456 is an anti-arthritic drug. Which of the following structure is not likely a metabolite? Let me check the chat box now. Okay, so the answers are given like number D and number A. Okay, now let me check which of the following is the correct one, number D or number A. Others also please participate even if your answer is wrong. But participation is very important. Otherwise, I won't understand if you are listening to me or not, if you are following the lecture or not. Okay, so please participate. Even if your answer is wrong, it doesn't matter. But please try to answer the questions. Okay, so now I will show you what is the correct answer for problem number 8. So the correct answer will be number A. So the structure A is not likely to be a metabolite of this drug L791456. So why it should not be with this what is the thing is called? So there is a drugs for drug stability. Okay, introducing susceptible metabolic group. So it is used to decrease the metabolic stability and drug lifetime, used as a drug for the drug which linger too long in the body and cause side effect. For and at groups known to the susceptible for phase 1 and phase 2 metabolic reaction. For example, this L7, so there is a drug which is L7872 787257, which is resistant to metabolism for the excessively long half life. Okay, but from this drug we have we get this L791456, which have a anchoring methyl group. If you look at here, here is a methyl group present. Okay, so this methyl group is introduced, which is introduced to increase the metabolic susceptibility for the because it is converted to this alcoholic and acidic group easily. So this will decrease the lifetime of the long lifetime of half life of the drug, and it will increase the potency of the drug. And if you look at the other structures carefully, so now why structure number A cannot be a possible metabolite? Because others, if you look at the other option carefully, so this chlorine group replaced by hydroxyl is not that much of feasible. Okay, but other look at the other possibilities, like this is the pyridine and nitrogen. Okay, so pyridines are converted to N oxides very readily. So you can see in number B we have an N oxide of this pyridine number with the ring, and in the case of number C we will have the N oxide of the this pyridine ring. Okay, so these are very easy reaction which happens very facile, and this conversion of this methyl group to carboxylic acid, it is also very metabolically susceptible process. But this metabolic this conversion from the chlorine to hydroxyl, it is not that much of facile. So structure number A will not be likely a metabolite of L791456. Okay, and here also the rationale of designing of the L791456 is given from the precursor L727 787257. okay, which is to increase the metabolic susceptibility of the drug. So now problem number 9, it says which of the following statements regarding neurotransmitter is not true. Your options are number A, they are secreted by the neuron cells. Number B, they are chemical messengers. Number C, they basically traverse through the blood to act on a distant target cell. Number D, they carry out a cascade of reaction. Which of the following statements regarding neurotransmitter is not true? Whenever there is a true, false or correct, incorrect kind of question, read the options very carefully because students are very likely to make mistake in this kind of questions. Okay. So it is said which of the following statements regarding neurotransmitters is not true. Your options: number A, they are secreted by neuron cells. Number B, they are chemical messengers. Number C, they basically traverse through the blood to act on a distant target cells. And number D, they carry out a cascade of reactions. Which of the following statements regarding neurotransmitters is not true? Let me check the chat box and then I will discuss it. Okay. The options, the answers are given. Number D, number A, and number C. Okay, number A and C. I think you are just confused a little bit. Okay. So let me check which of the following will be the correct answer. So the correct answer will be number C. They basically traverse through blood to act on a distant target cell. It is not true. So now let us understand what are neurotransmitters. So neurotransmitters are chemical substances released by the electrical impulses into the synaptic cleft from the synaptic vesicles or presynaptic membrane. Then it diffuses to the postsynaptic membrane, binds to and activates the receptors present, leading to initiation of a new electrical signals or inhibition of postsynaptic neuron. So the examples of neurotransmitters are adrenaline, which are called fight or flight. They're produced in a stressful situation, increases the heart rate and blood flow, leading to the physical boost and heightened awareness. Okay, then where we have a GABA, which is a GABA. butyric acid, and this comes the flooding nerves on the central nervous system. High levels of improved focus, low levels can cause anxiety, also contributes to the motor control and vision. We have noradrenaline, which is responsible for the concentration, affects the attention and responding actions in the brain, contracts the blood vessels, increasing the blood flow. Acetylcholine for learning, involved in thought, learning and memory, activates the muscle action of the body, or also associated with the attention and awakening. Dopamine, which is for the pleasure, feelings of pleasure, also addiction, movement of motivation, movement and motivation. People repeat behaviors to that lead to the dopamine release. And glutamate for memory, most common neurotransmitter involved in the learning and memory, regulates the development and creation of the nerve contracts. And we have serotonin for mood, it contributes to the well-being or happiness, helps to sleep cycle and digestive system regulation affected by the exercise and light exposure. And endorphins, which for euphoria, released during the exercise, excitement, and is producing the well-being and euphoria and reducing the pain. So what are the differences between hormones and neurotransmitters? Hormones are released by the endocrine glands, okay, and neurotransmitters are released by the nerve cells. Hormones travel through the blood to act on a distant target cell, but neurotransmitters travel across the synapse to act on the nearby cells. The hormones affect the effects of hormones can be slower to build but longer lasting. The effects of neurotransmitter can be fast to act but shorter lasting. Hormones are a part of the endocrine system, neurotransmitters are part of the nervous system. Okay, so these are the differences between the hormones and neurotransmitters. So now I will move toward the next problem. So problem number 10 says molecules which are formed by reducing the peptide nature of lead molecules are known as number A peptidomimetic, number B oligonucleotide, number C prototype, and number D neurotransmitter. What are the molecules formed by reducing the peptide nature in the lead molecule is known as? Your options are peptidomimetic, oligonucleotide, prototype, and neurotransmitters. I request all of you to participate and answer which of the following statements will be, which of the following option will be correct for the molecules which are formed by reducing peptide nature in the lead molecule. Let me check the chat box. Okay. Number A, number C. I think it is from same person. Okay, so I have two different options, number A and number C. Now let me check which of the following will be the correct one. So the correct answer will be number A, peptidomimetic. So peptidomimetics, which is incorporating the conformational constant locally or globally. What is the peptidomimetics? These are the molecules with significantly reduced peptide character that mimic the bioactive conformation of peptides and thus retain the ability to interact with the biological target and cause the same biological effect. Small protein-like chain designed to mimic a peptide arise either from modification of an existing peptide or by designing similar system that mimic peptide, for example, peptides or beta peptides. So peptidomimetics are molecules which are look or like or feel like or behave like peptides. Okay, so because peptides continue to be important lead molecules, a lot of medicinal chemistry has worked around peptides, and because peptides as receptor ligands and enzyme substrates are very commonly found. Therefore, we do not want to give up on peptides. So therefore, we will want to be able to deliver the peptide or peptide-like molecule which is known as peptidomimetic. So consequently, drugs which are designed from these lead compounds which what are peptide-like in nature have taken forward. So the peptide, the pharmacokinetic of the first generation drug of using peptides are many unsatisfactory, and the number of strategies that has evolved to improve the bioavailability to achieve the higher level or acceptable level of these molecules in the blood supply. So therefore, disguising or reducing peptide nature of the lead compound to generate a structure which are more easily absorbed in the GI tract and more resistant to digestive and metabolic enzymes is highly desirable, and these molecules are known as peptidomimetics. Okay, so the option number A, peptidomimetic, was the correct answer. These peptidomimetic molecules are formed by reducing the peptide nature of lead molecule. Because the peptide nature of the lead molecules, that is not very good for the pharmacokinetic profile of a particular drug due to the lower bioavailability. But the peptidomimetics, they have improved biological action and improved bioavailability owing to their structures. Okay. So now next problem number 11 says what of the following, which of the following is a peptidomimetic formation technique? Your option are number A, pegylation, number B, replacing L amino acid with D, number C, N methylation, and number D, all of the above. Which of the following is a peptidomimetic formation technique? Pegylation, replacing L amino acid with D, N methylation, and all of the above. In last slide, I have discussed what is a peptidomimetic. So now there are several strategies which are used for the formation of a peptidomimetic. So which of the following in given options is the correct answer? Number A, pegylation, number B, replacing L amino acid with D, number C, N methylation, and number D, all of the above. Let me check the chat box now. Okay, so there are options number B and number A. Okay, let me check which of the following will be the correct one. So the correct answer will be number D. All of the above methods can be used for the peptidomimetic formation. So, disguising or reducing the peptide nature of the lead compound to generate a structure which is more easily absorbed from the gastrointestinal tract and is more resistant to digestive and metabolic enzymes, such analogs are known as peptidomimetics. It has been found that linking the polymer polyethylene glycol, which is known as PEG, to the protein can increase the later stability and the solubility, as well as decreasing the likelihood of an immune response. Pegylation, as it is called, also prevents the removal of small proteins from the blood supply by the kidneys or the reticulometrical system. The increased size of the pegylated protein means it is not filtered by the kidney by nephron and remains in the blood supply. PEG molecules surrounding the protein can be viewed as a kind of hydrophilic polymeric shield which both protects and disguises the protein. The PEG polymer has also added advantage in that it shows the little toxicity. So the methyl group acts as a steric shield that prevents the important hydrogen bonding interaction supposed to plate between the so if there is a N-methylation, in case of N-methylation, the methyl group acts as a steric shield to prevent the important hydrogen bonding interaction that is supposed to take place between NH of the original amide or peptide and the active site of peptide is enzyme. So therefore, now the steric shield is present, the rate of hydrolysis significantly go down. So N-methylation is also one way to generate the amide like to like or to retain the peptide of the amide as a drug. Another unrelated to the completely strategy is to flip the enantiomer we use. So for example, if we replace the L enantiomer that is normally present in protein with the D enantiomer, then what happens is that it makes the relative orientation unrecognizable to digestive and metabolic enzyme, especially if the side chain is involved in the interaction. So here, for example, if you have L amino acid, then you convert two L amino acids, but if you convert one of this is like as to D amino acid, it is possible that it may not get hydrolyzed as well as the original molecule. Okay. So the three processes, pegylation replacing L-amino acid with D and the N-methylation, all of the above can be used as a peptidomimetic formation technique. So now next problem, problem number 12 says oligonucleotides suffer disadvantages as drugs since they are susceptible to the enzyme catalyzed degradation. Which of the following enzyme is mainly responsible for this? Your options are number A peptidase, number B nuclease, number C lipase and number D oxidase. Which of the following enzyme is mainly responsible for the disadvantages that oligonucleotide suffers as a drug? Oligonucleotides suffers disadvantages as drugs since they are susceptible to the enzyme catalyzed degradation. Which of the enzyme is mainly responsible for this? Peptidases, nucleases, lipases or oxidases? Okay, let me check the chat box now. Okay, number A, number B. Okay, let me check which of the following will be the correct one. So the correct answer, the enzyme which is responsible for the degradation of the oligonucleotides is number B, nucleases. So nuclease enzymes degrade the nucleic acids by breaking the phosphodiester bond that holds the nucleotides together. Restriction enzymes are a good example of the endonucleases which cut within the DNA strand. A second group of nucleases which degrade the DNA from the termini of molecule are known as the exonucleases. Like this is the example, this is unmodified DNA or RNA, and here we have the phosphodiester bond, but the backbone modification happens. So like your phosphorthionate like this, and sometimes there is a this is the backbone modification, so that the nucleases cannot actually break the phosphodiester bond, so that the modification are required, such as the conversion of that particular phosphodiester bond to the phosphothionate bonds, okay, or phosphoamidase bond or morpholino bonds, like this, okay. So these are the modifications. So remember, nucleases are the enzymes which are responsible for the degradation of oligonucleotides. Now problem number 13, it says that why should the addition of an alcohol or phenol group to a drug decrease its duration of action? Your options are number A, it acts as a polar handle of conjugation reaction. The conjugates are excreted more quickly. Number B, it increases the polarity of the drug, reduces the amount of drug absorbed. Number C, it reacts with the proteins in the body such that the drug is irreversibly linked to the proteins by a covalent bond. And number D, it acts as an electron withdrawing group and affects the binding strength of the important binding groups. So why should the addition of an alcohol or phenolic group to a drug decrease its duration of action? Your options are number A, it acts as a polar handle for conjugation reaction and the conjugates are excreted more quickly. Number B, it increases the polarity of the drug, reduces the amount of drug absorbed. Number C, it reacts with the proteins in the body such that the drug is irreversibly linked to the proteins by the covalent bond. And number D, it acts as an electron withdrawing group and affect the binding strength of the important binding groups. Let me check the chat box now. Okay, so number A is the given answer till now. Okay, let me show you the answer because we have till few questions which we need to discuss. So the correct answer will be yes, it is number A. It acts as a polar handle for the conjugation reaction. The conjugates are excreted more quickly. So as we will see, during the phase 1 and phase 2 metabolism, what happens, like we have drugs and then we have the lipophilic drugs which after oxidation forms that particular and they form this electrophiles, okay, and also by the hydrolysis reduction they form the nucleophiles. These nucleophiles will form, in case of the phase 2 metabolism, this will form the hydrophilic portions like the sulfuric acid, then acylation and glucuronidation, okay, and glutathione conjugation will also make this drug hydrophilic. So this phase 1 and phase 2 or phase 2 phase 2 metabolism, these are conjugation reactions. So what happens in case this conjugation conjugation reaction, these following groups are involved, like sulfate, alpha glucuronic acid, glutathiones, okay, the methyl glycine, glutamine, and acetyl. These groups are transferred to appropriate conjugation enzyme on the drugs or other metabolite. Okay, so this phenolic OH or alcohol group, they act as a polar handle for this kind of conjugation reactions with these groups, okay, and these conjugates are excreted more quickly. That's why the phenolic OH group or alcohol group, they decrease the duration of action of a particular drug. So now next problem. Problem number 14 says CGP52411 is a protein kinase inhibitor for which drug metabolism studies show that the para-hydroxylation occurs to produce inactive metabolites. What strategy can be taken to protect this compound from metabolism? Your options are number A stereoelectronic shield, number B electronic shield, number C steric shield, and number D alkyl shield. CGP52411, this drug is a kinase inhibitor for which metabolism studies show that para-hydroxylation of this group, this phenyl rings, produce inactive metabolite. What strategy can be taken to protect this compound from metabolism? Stereoelectronic shield, electronic shield, steric shield, or alkyl shield? Let me know in the chat box, A, B, C, or D, which of the following you think is the correct answer. Let me check the chat box now, then I will discuss it. Okay, so one answer is given, is the steric shield, and then I have another answer which says it is number D. Okay, so number C and number D is the given answers. Okay, let me check if steric shield is the correct answer or the alkyl shield. So the correct answer will be number C, steric shield. Very good. So why steric shield? So let me show. So this CGP52411, it is a useful inhibitor of protein kinase enzyme. Studies on the structure-activity relationship demonstrate that substituent on the aromatic ring such as chlorine, methyl, or hydroxide are bad for activity. Drug metabolism studies show that para-hydroxylation occurs to produce inactive metabolites. So how would you modify the structure to protect the metabolism? The answer will be the steric shield. Okay. So the steric shield strategy, what happens for the steric shield strategy? So this para position is important. Okay, this para position, it is susceptible for the hydroxylation, right? So if we add methyl groups in this position, let me just draw it for your understanding. This para position, it is susceptible for hydroxylation. Okay, but if we add methyl groups here, like here, here, and here, so what happens? Because of this methyl group, this steric bulk of the methyl group, this position is now not available for the hydroxylation. So now this strategy, as I have already mentioned previously also, it is known as a steric shield, not an alkyl shield. I think you have you have just answered it as alkyl shield for, because I am adding the alkyl group. Okay, it may seem like it can be act as an alkyl shield, but alkyl shield is not the term which is used for in case of this kind of strategy. So the steric shield strategy, because this position is to attack, what we could do is, like, we could add one or two methyl groups around this carbon and this present as a steric shield to be for it to be oxidized. Okay, so of course this problem now with this approach is, then you can, is interesting that the hydrophobicity of the molecule is adding methyl groups, so that we could just do this. And first we could so first we could just add one methyl group at meta position to see how the susceptible to oxidation, and follow up to second if necessary. Okay, so steric shield was the correct answer. So now problem number 50 says why morpholine and pyridine rings are added to a drug molecules. Your options are number A, to improve the hydrophobicity, number B, to improve the water solubility, number C, to increase the molecular weight, and number D, all of the above. Why morpholine and pyridine rings are added to drug molecules? These morpholine and pyridine rings are proven to be very important for the drug's activity. So why be these to improve the drug's activity? Sorry. So why these rings are very important? Because number A, to improve the hydrophobicity, number B, to improve the water solubility, number C, to increase the molecular weight, and number D, all of the above. Which of the following will be the correct answer? Let me know in the chat box, A, B, C, or D, which of the following you think is the correct answer. Let me check the chat box now. Okay. Number B, increase solubility, number B. So number B is the given answer. Okay, let me check. Number B is the correct one. So the correct answer will be, yes, number B, to improve water solubility. So this is the structure of morpholine. This is the structure of pyridine, right? So The first question explain why the morpholin and pyridine is added to the drug molecule. So it is many times we find that the drug molecule is actually quite poorly soluble. So in order to improve the solubility, one needs to add polar groups that can be hydrophilic in nature, but they are not going to be dramatically alter the properties of a drug. So here in nitrogen containing heterocycle are such that morpholin and pyridine are quite often used. So the nitrogen is polar and definitely increase the polarity, and in some cases protonated form can also be isolated as water soluble salts. So here, for example, if you connect it with this nitrogen and then you can sort this whole molecule, morpholin is quite polar, and here in case pyridine, you can make pyridinium salt, which is actually isolated as pyridinium salt. So if a polar group is added in order to increase solubility, it is preferable to add the molecule in such a way that it is exposed to surrounding water. Okay, so therefore, because it is exposed to surrounding water, the drug, it is bound to the target site, does not have sort of dissolve it before the binding to the active site. So this is an important criterion when we are trying to add more functional group to the drug. Okay, so now problem number 16, it says that why lidocaine has improved bioactivity than procaine. So this is the structure of the procaine and this is the structure of lidocaine. Your options are number A steric hindrance, number B electronic stabilization, number C both, and number D none. So why lidocaine has improved bioactivity than procaine? Steric hindrance, electronic stabilization, both, or none? Why lidocaine has improved bioactivity than procaine? Let me check the chat box. Okay, number A and number D are the given answer. Okay, let me check if number A is the correct one or number D. Okay, so the correct answer will be number C, both. Electronic stabilization and steric hindrance both are responsible for the improved bioactivity of lidocaine than procaine. So steric hindrance and electronic stabilization have often been used together to stabilize labile groups. For example, procaine is a good but short-lasting local anesthetic because of the ester group, okay, which is quickly hydrolyzed. There is a ester group here. This ester group is quickly hydrolyzed. That's why procaine, which is a good anesthetic, but it is a very short-lasting. The analog lidocaine was developed then. Okay, the changing ester group to less reactive amide group reduces the susceptibility of the chemical hydrolysis. Furthermore, the presence of two methyl groups in the aromatic ring helps to shield the carbonyl group from attack of the nucleophile or the enzyme. So as you can see here, not only the more susceptible, electronically susceptible ester group is replaced by the amide group, which is more stable, but also two methyl groups are added on the vicinity of the amide linkage, so that this particular group, this particular carbonyl carbon can be protected from the incoming nucleophile, which improves the bioactivity of the drug. So steric hindrance or the steric shield and also electronic stabilization both has taken part for improving the bioactivity of lidocaine from the precursor procaine. Okay, so number C is the correct answer. So now next problem. Problem number 17 says the correct statement among the following regarding the antihypertensive drug enalapril is: number A, it is an irreversible inhibitor of angiotensin converting enzyme. Number B, it is a prodrug. Number C, it has got poor bioavailability, and number D, it is a transition state analog inhibitor of the angiotensin converting enzyme. Which of the following statement for regarding the antihypertensive drug enalapril is the correct one? Again, I will repeat the options. Number A, it is an irreversible inhibitor of angiotensin converting enzyme. Number B, it is a prodrug. Number C, it has got poor bioavailability. And number D, it is a transition state analog inhibitor of angiotensin converting enzyme. Now let me check the chat box. Number B is the given answer. Okay, let me check. So the correct answer or the correct option will be yes, number B, it is a prodrug. Phenanapril is an angiotensin converting enzyme, ACE inhibitor, which is a prodrug which can be hydrolyzed by esterases to the active enaraprilate. The mechanism of action is enaraprilate competes with the angiotensin 1 for binding at the angiotensin converting enzyme, blocking the conversion of angiotensin 1 to angiotensin 2. As angiotensin 2 is a vasoconstrictor, the negative feedback mediator for the renin activity. Lower concentration result in decrease in blood pressure. Enalapril may also act on kinase 2 and degrades the vasodilator bradykinin. So this is the enalapril. It is a prodrug which can cross the membrane, and it is the enalaprelate which is the antihypertensive agent. Okay, and this okay, I think there is a I should say. So yeah, enalapril is a ester. Okay, and enalaprelate, it is the antihypertensive agent, which is the active form for this prodrug ester. So here the ester is converted by the esterases to the acid formation, and this is the active form. Okay, the enalaprelate. So yeah, number B was the correct one. Now problem number 18 says which enzyme is responsible for the SCID. Okay, severe combined immunodeficiency syndrome. Which enzyme is responsible for severe combined immunodeficiency syndrome, SCID? Your options are number A, catechol dehydrogenase, number B, cytochrome C oxidase, number C, adenosine deaminase, and number D, adenylate deaminase. Which of the following enzyme is responsible for SCID? Catechol dehydrogenase, cytochrome C oxidase, adenosine deaminase, or adenylate deaminase? Let me check the chat box now. Okay, number C is the given answer. Let me check if it is the correct one or not. So yeah, the correct answer will be number C, adenosine deaminase. So adenosine, this is the adenosine. Okay, and adenosine, okay, let me just set pointer. So this is the structure of adenosine, okay, and for by hydrolysis it forms the inosine and the ammonia. Okay, the hydrolysis here in this position it forms the inosine and ammonia. So severe combined immunodeficiency or SCID, it consists of a group of genetic disorder characterized by the block of the a block in the T lymphocyte differentiation that is variably associated with abnormal development of other lymphocyte lineages such as B or NK lymphocytes, and more rarely of the myeloid lineage. It is a genetic disorder in which both arms, B cell and T cells of the adaptive immune system are impaired due to the defect in one several possible gene. SCID, a severe form of the heritable immunodeficiency. The overall frequency is estimated 1 in 70,000 to 1 lakh births. Okay, so this is the deoxyadenosine. Okay, and this adenosine deaminase, what it happened, it forms the deoxyinosine here, and it forms the uric acid. So this particular deoxyadenosine, this is a triphosphate of the deoxyadenosine triphosphate. Okay, so now problem number 19, it says that there are several strategies for stabilizing susceptible bonds in peptides. Which of the following is not used in this respect? Okay. Number A, replace the peptide link with a stable functional group. Number B, adding an N-methyl group in peptide link. Number C, using an unnatural amino acid. Number D, replacing D amino acid with L amino acid. There are several strategies for stabilizing susceptible bonds in peptides. Which of the following is not used in this respect? Number A, replace the peptide link with a stable functional group. Number B, adding an N-methyl group to the peptide link. Number C, using an unnatural amino acid. And number D, replacing D amino acid with L amino acid. Which of the following is not used for stabilizing the susceptible bonds in peptides? Let me know in the chat box which of the following you think is not used for the stabilizing purpose of the susceptible bonds in peptides. Let me check the chat box now. Okay, number D, number C is the given answers. Okay, let me check. So the correct answer will be Number D, replacing D amino acid with L amino acid is not used. This strategy is not used for stabilizing a susceptible bond in peptides. Now let us look at the differences between L amino acid and D amino acid. So L amino acid is stereoisomer of the particular amino acid whose amine group is on the left side of the Fischer projection, and D amino acids are the stereoisomer where the amine group is the right side of the Fischer projection. The L amino acid can rotate plane polarized light counterclockwise in a process called levorotation, and the D-amino acid can rotate the plane of polarized light clockwise in a process of dextrotation. It is superseded by R notation, and the D-amino acid are superseded by S notation. The L-amino acids are used to by the cell to produce the proteins, and the D-amino acids occur in the cell wall of the bacteria. Okay, so these are the differences between the L-amino acid and D-amino acids. So now next problem, this is the last problem of today's session. It says that candoxatril is a ester prodrug of candoxatrelate which inhibits the protease enzyme. What are the following statements is incorrect? Number A, hydrolysis of the ester reveals a carboxylic acid group on candoxatrelate. Number B, the bicyclic leaving group is non-toxic. Number C, the parent drug can't be administered orally, whereas the ester can. Number D, the bicyclic system is electron donating and speeds up the rate of the ester hydrolysis. Which of the following statements is incorrect regarding this candoxatril? Number A, hydrolysis of the ester reveals a carboxylic acid group on candoxatrelate. Number B, bicyclic leaving group is non-toxic. Number C, the parent drug can be administered orally, whereas the ester can. And number D, the bicyclic system is electron donating and speeds up the rate of the ester hydrolysis. Okay, let me check the chat box. Okay, number D and number C again, these two are given answers. Okay, let me check which of the following is the correct one. So, number D is the correct answer. Okay, the bicyclic system is electron donating and speeds up the rate of the ester hydrolysis. It is, the statement is the incorrect. Okay, now let me show you. So, in prodrugs to improve membrane permeability, the example is candoxatril for candoxatrelate, which is a protease inhibitor. This is the structure of candoxatrelate, and this is the candoxatril. Okay, there is a 5-indienyl group present here. Okay, so varying the ester varies the rate of hydrolysis. The electron withdrawing groups increase the hydrolysis, and the leaving group is non-toxic here. Okay, so option number D was the incorrect statement. So yeah, today it was the last question of today's session. So today we discussed about the prodrug design. We discussed several strategies of the prodrug design and several strategies to improve the drug stability. So all of you have participated very well and answered the questions very good. So right now I will just share my email ID here. Okay, in the chat box. Just let me give a minute. So this is my email ID. If you have any doubt, any query regarding today's class, and also if you have any doubt, any query regarding any topic of the course medicinal chemistry till the week 9, you can feel free to mail me on this email ID. Okay, I will try to answer this email ID, and try to answer your queries in this from this email ID. Also, you can just write your query on the discussion forum for the NPTEL course of medicinal chemistry, which you will find on the SWAM portal. So today it was our week 8 live session, and this kind of live session we will have every week on Tuesdays from 6 p.m. to 8 p.m. I will share this PowerPoint presentation and also the lecture video which I have recorded for the class. Okay, just you can check there. If you have any, if you have missed anything, you can just check there. So very good. That's two of you have responded very well and answered most of the questions very clearly and also very correctly. So very good. So just this kind of participation I require from your side, so that I can understand if you are following or not. So if you have any problem, any doubt, any query regarding any topic of medicinal chemistry, please just drop an email to me on this email ID. So today I will just close the session. Let's meet on the next Tuesday from this time 6 p.m. to 8 p.m. and the joining Gmeet ID will be same, which is this one. Okay. So thank you all. I will just leave the session and I will just close the session for today.